So for those of you that are not familiar with it, Duchenne muscular dystrophy it occurs due to a mutation in the DMD gene, which means that patients lack the protein dystrophin. So dystrophin is normally located around all the muscle fibers, and this is a section of muscle from a DMD patient that has no dystrophin. So dystrophin is a part of a huge complex, the dystrophin-associated protein complex, that spans the plasma membrane of the muscle fiber. And if dystrophin is lost, much of the complex is lost as well. And as a consequence, the muscle fibers degenerate. So regeneration does occur, but of course the regenerated fibers still don't contain dystrophin, and then they go on to break down again. And the regeneration eventually fails to keep up. Here are some regenerated muscle fibers. And in the end, there's lots of fibrous connective tissue. And at a very late stage, there are very few muscle fibers, and the muscles nearly all replaced by fat or connective tissue. So there are muscle stem cells, but the question is, are the muscle stem cells directly affected by the genetic mutation or indirectly affected? The stem cells are okay, but the environment affects their function. And we are very lucky in that there's a naturally occurring mouse model, that the MDX mouse, that also lacks dystrophin, that many of us have used in our research. So really it's quite, the whole concept is quite simple. You can take cells from a donor, grow them in culture, transplant them into a patient, or you can take the patient's own stem cells and then either insert or repair the defective gene and transplant them back. So ideally, the transplanted stem cells will expand within the muscle and then differentiate into muscle fibers. And muscle stem cells differentiate by fusing either with each other to form a muscle fiber purely of donor origin, or they repair segments of damaged muscle fiber. So within the regenerated muscle fibers, dystrophin should be expressed, and this restored protein will prevent the muscle fiber from undergoing any more breakdown. And ideally, the transplanted cells will also give rise to more functional stem cells, which will contribute to long-term muscle maintenance and repair and regeneration. So we've been using a model system to quantify the contribution of different sorts of human stem cells to muscle regeneration. So we use a host mouse, which is immunodeficient, and most of the work I'm going to be talking about today is using the dystrophin-deficient MDX mouse. And we've used different sorts of cells derived from human skeletal muscle and also induced pluripotent stem cells. And these are either derived from normal donors or DMD donors, and the DMD cells may be genetically corrected before transplantation. So we transplant them intramuscularly, and then we look to see whether they've contributed to muscle regeneration by looking for, by using antibodies that are specific to human spectrin to identify muscle fibers, to human lamin AC to identify nuclei of human origin. We can also see if dystrophin's been restored. We can also see whether the transplanted cells have given rise to muscle stem cells or satellite cells by using a combination of an antibody identifying the satellite cell PAX7 in combination with an antibody to identify human nuclei. So this is a satellite cell of human origin within the transplanted muscle. We can also see if these satellite cells of human origin are functional by transplanting the cells into the muscle, leaving them to regenerate, and then injuring the muscle and looking to see if they're newly regenerated muscle fibers, these green ones here that also have human origin, expressing human spectrin. So this is evidence that our transplanted cells are functional. They made satellite cells, and these muscle stem cells of donor origin were functional. They can contribute to muscle regeneration after further injury. So in our preclinical work using this cell transplantation model, we've shown that many different cell types derived from skeletal muscle can contribute to muscle regeneration, and many of them can also contribute to functional satellite cells. But our work, we've only been successful after injecting the cells intramuscularly, and only if the muscles have been injured before transplanting the cells. So this looks like a lot of muscle of donor origin, but in fact it's about 150 fibers, and this is in a muscle which contains 1,500 muscle fibers. So it's only a small percentage of the whole muscle is dystrophin positive. The other problem is that many of these cells lose their capacity to contribute to muscle regeneration after they've been expanded to large numbers in tissue culture. So this just summarizes work done by lots of different labs looking at what cells do contribute to skeletal muscle regeneration. And the ones in black really do work, and they're either the muscle stem cells, the satellite cells, or their progeny myoblasts. They're also cells from skeletal muscle associated with blood vessels, pericytes, or mesoangioblasts. Also cells present in skeletal muscle that express the antigen CD133, and these all contribute to muscle regeneration, but they all have limitations. So ideally, we'd want to prepare our stem cells without having to do an invasive muscle biopsy, something easily obtainable from blood or skin. And we want to make sure that the cells can retain their phenotype after expansion in culture. We also want to make sure that they make muscle after systemic delivery, and they give rise to large amounts of muscle and also to functional muscle stem cells. So we've been focusing on autologous stem cells. So the advantage of these is that they'd be less likely to elicit an immune reaction than donor stem cells. So the disadvantage is that they'd have to be prepared from every patient. And we've been looking at lentivirally transducing them with a dystrophin construct. And the advantage of this is we could use the same construct for all patients, regardless of the patient mutation. We've also been looking at genetically correcting stem cells. And of course, because the DMD patients have got a range of different mutations, this would have to be patient specific. So the advantages of lentivirus is that it transfects both cells that are proliferating and also non-proliferating cells, for example, post-mitotic muscle fibers or quiescent stem cells. It integrates, so it's heritable or daughter cells that express the transgene. It's safe, it's been used in clinical trials for different conditions. But it's got quite a limited cloning capacity, which is a disadvantage. And the promoter driving the dystrophin expression, we've got to consider it'll have to be a small promoter to fit into the viral vector. And also the time and extent of expression during the muscle differentiation is, of course, is something we've got to consider. So, as I said, the lentiviral cloning capacity was initially thought not to be able to incorporate the full-length dystrophin cDNA. Dystrophin's very large. So we looked at smaller dystrophin constructs, which retain some, if not all, of the functional elements of full-length dystrophin. And we used these constructs to make lentiviral vectors. And in these experiments, the dystrophin was driven by an ubiquitous SFFE promoter. So we transduced pericytes from DMD patients with these dystrophin constructs, transplanted the transduced cells into our intramuscularly into our host mice, and we found that the cells contributed to muscle fibers expressing dystrophin. And interestingly, these dystrophin expressing muscle fibers also expressed alpha-sarcoglycan, which is a member of the dystrophin associated complex. So this dystrophin is functional in that it's reassembled other members of the dystrophin-associated complex. So just after this, John Counsell in our group showed that you could actually incorporate full-length dystrophin into a lentiviral vector. So this top panel here shows a myoblast from a DMD patient, and when they differentiate into myotubes in culture, they don't express dystrophin. The transduced stem cells with the full-length dystrophin, when they differentiate into myotubes, they express dystrophin. And this Western blot shows that the dystrophin is full-length, so it's the expected size. So these transduced myoblasts were transplanted intramuscularly into our host mice, and they gave rise to muscle fibers of donor origin expressing dystrophin. So this looks very promising, because if we can put the full-length dystrophin in, it will be fully functional, and the muscle should then be muscle fibers then should have restored function. So currently, we're looking at cells that can be easily obtained without having to do a muscle biopsy, that can be expanded and manipulated in culture. And we're looking at induced pluripotent stem cells, which either derive from donors or from DMD patients. And we're looking to genetically correct them or lentivirally transduce them, and looking with our mouse model system whether they contribute to muscle regeneration. So we've been using a protocol derived by Olivier Pourquery's lab to make coax induced pluripotent stem cells to undergo myogenic differentiation. And these are from some control-induced pluripotent stem cells, and they express PAX7, so they're satellite cell-like, and they can differentiate in culture into multinucleated myotubes. And when we transplanted these myogenic cells derived from normal controls into our mouse muscles, they gave rise to muscle fibers of donor origin, and these muscle fibers expressed dystrophin. So this is quite promising. So our ongoing work, we're focusing on iPSCs derived from different DMD patients. And we're either gene editing them with CRISPR-Cas9 in collaboration with young Yao Lin at Queen Mary University of London, or we're lentivirally transducing them with the full-length dystrophin, and we're using full-length codon-optimized dystrophin in collaboration with George Dixon and Linda Popplewell at the Royal Holloway. We're also trying to look at the best promoter to use. It's got to be small and it's got to have the correct expression profile, because if dystrophin is expressed at the wrong time during a myogenic proliferation and differentiation, it can have an adverse effect. Also, John Council is looking at clinically safer lentiviral vector construction. And we're also part of a collaborative international piece of work to try and identify the best human stem cell for future clinical applications. So there's still challenges. Getting enough cells to treat all of the muscles body-wide is a huge challenge. Being able to deliver them systemically via the blood system and have them make enough muscle in downstream muscles is a challenge. The host muscle environment might also affect the muscle stem cell function. And a real challenge is getting enough dystrophin to be of functional benefit to the muscle, because other work has shown that you need all of the muscle fibers in a muscle to have between 10 and 30% of 10 and 30% of normal dystrophin levels to improve muscle function. And other challenges are that, of course, the heart and also the brain are affected in DMD patients. So the work that I've very briefly gone through today, we're extremely grateful for all the people that have funded this work over the years, especially in all the charities. And these are the collaborators that have contributed to this part of the work. And I really need to acknowledge Jin Hong Meng, the postdoc in our group, who has led this work over many years now. And also the MRC Centre for Neuromuscular Diseases Biobank, Pier Paola Alla, who's provided us with the cells, with the human cells for this work. So thank you very much.